Good day, students. Today is our 12th session. The title of the session is Fungi Cultivation and Medical Importance. So, we are going to speak how to culture fungi in the laboratory. Cultivation of fungi, this constitutes the two types of fungi, molds and yeast. This can be grown in a medium, culture media, in the laboratory, the same as bacteria. The media used for fungi, they are just modified with the addition of certain ingredients that inhibit the growth of other microbes, because usually the media for the fungi is very rich in nutrients. And as you know, fungi are mostly saprophytic and parasitic, so they tend to utilize as much of the nutrients as possible. That's why they require very high nutritive culture media with very high nutritive value. So we need to add certain inhibitors to the media to prevent other microbes from growing, because this type of media is also suitable for bacteria and other non-required fungi. So in this case, we add antibiotics to prevent bacterial growth, and we can add dyes which change the pH of the medium, or we can add very high concentration of sugars, also inhibit other types of fungi and other type of microbes. And even we can lower the pH of the medium to below 5 or 3 pH, because most of the fungi can grow between the range of 3 to 5 pH, unlike bacteria, which most of them they prefer the pH to be between 5 and 7, or even alkaline pH. There are two types of media, three types of basic media. Natural, when we say natural, that means we can use certain types of plants, and mostly we use carrot plugs and potato slices. If you put a potato slice in a petri dish and you add a fungi, you will find after incubation that the fungi started to grow. And this you can even notice it in the kitchen. If you leave certain plants or certain vegetables outside, then this can be a good medium for fungi in the air to grow. So this we call them natural media. The second type is the dehydrated media, and this is similar to the media of the bacteria, but it's a different type. That means it's found in powder, and you need to prepare it and sterilize it, the same as the process of the bacterial media, to sterilize it and powder it either in the tube or in the petri dish. The one that is mostly used is the subrowed dextrose agar. The third type is the synthetic media, and this is prepared by certain companies, and it is found ready-made in the petri dishes. And this type is the potato dextrose agar. This is a very rich media in nutritive value, and the fungi, they tend to produce mycelium very fast on this media, and they lose the sporulation, the spores, because they have all the available nutrients in the media. So if you want to study the spores of these fungi, you need to plate them or culture them on, we call them a starvation media, that is containing less nutritive value. And this is a type of this medium is the potato carrot agar. That one is potato dextrose agar, which is very rich in sugar, and the other type is potato carrot agar. And this again, it is a synthetic medium, but it is suitable for the fungi, especially the hyphae fungi or the filamentous fungi, to grow and produce the spores. So if you want to study the spores of the fungi, you need to plate it or culture it on a starvation medium like potato, carrot, agar. There are certain requirements for the growth of the fungi, like the bacteria. So we have the temperature. The majority of filamentous fungi are mesophilic. That means they grow in the range of 10 to 35 degrees centigrade. Generally, fungi, they can grow in the laboratory between 20 and 25. This is the optimum temperature or the suitable temperature. But the range is from 10 to 35 or 10 to 15 to 30 degrees centigrade. Light, many species of fungi, they prefer to grow in the dark. But there are others which prefer to grow in the daylight, like the fungi that affect or infect plants, the stems and leaves of the plants. They prefer light to grow, and they produce mycelium in the when they infect these plants. The third type of bacteria of fungi, they like to grow in the presence of ultraviolet light. And as you know, ultraviolet light, they have different wavelengths range or a wavelength range from 300 to 380 nanometer. So if this type of fungi, they require ultraviolet, you need to prepare ultraviolet, and this is available in the microbiology lab for these to incubate this in ultraviolet incubators. And it's better to grow or culture this type of fungi in plastic petri dishes, because the glass is prevents the penetration or transfer of the ultraviolet light. The third requirement is oxygen. All fungi are usually aerobic, they require oxygen, but there are certain types of fungi that require more concentration of oxygen, especially the um, the fungi that are found in the aquatic, we call them aquatic fungi in the water environment, like sea, oceans, and so on, or ponds. They require more oxygen. So if you want to grow them in the lab, you need to provide an aeration system. Aeration system, that means adding more oxygen to the environment or the medium, and usually they grow in liquid medium. To add more oxygen by a certain type of equipment in the lab, you call it bubbling air, and then this will require more oxygen, will provide more oxygen to the fungi to grow. The fourth requirement is the pH. Usually the fungi grow in the pH range from 3 to 7, but they prefer usually 3 to 5. And more, part of them, they can grow below 3 in the pH 2. So most of the fungi, they prefer acidic medium. Water, another requirement. As we already spoke before about fungi, they prefer the moist environment. So they prefer the environment that contains higher amount of water or humidity. So water, all fungi, they need water for growth, and the level or the percentage of water differs from one fungi to the other. But all of them, they require water presence in the medium, or what we call humidity. When you grow them in the incubator, you need to provide humidity, not dry incubator. That's why they require water as a moisture environment for their growth. After we grow the fungi in the medium, on the medium in the laboratory, we need to identify these fungi. So, identification of fungi is through two, two processes. The first process is the macroscopic examination of fungi colonies on the petri dish. So when we say macroscopic, that means by the naked eye, you can see the shape of the shape, the color, the elevation, the margins of the colonies in on the petri dish. Usually, fungi they have very characteristic colonies which differ from the bacteria. They have large colonies, and usually they are colored, and they have different margins. So this is a characteristic of the different genus of fungi. So for macroscopic examination, you can note the colony texture, you can note the colony color in the plate. So for the yeast, usually the colonies they represent those of the bacteria, but you can further identify it by the second step, which is the microscopic identification. So in the colony, in the macroscopic examination, you can note this is a key. This figure is a key for colony morphology. So you can note the form. Is it circular? Is it irregular? Is it filamentous? Is it rhizoid? The elevation, you can note the elevation, and you can note the margins. These are all characteristics of the fungi um, colonies on the medium, and these differ from one genus to the other. The second step, we're going to speak about the microscopic examination of fungi. For the microscopic examination of fungi, we use one of the three methods. The first method, which is commonly used in the microbiology laboratory, is what we call lactophenol cotton blue teased mount. This is a type of wet preparation, and you already know what is wet preparation because you did it in the first microbiology practical, how to prepare a wet preparation. So this is a wet preparation of the fungal culture where you take a drop of water on the slide, and then you take part of the culture on the medium onto this water, and then you cover it with a cover slip and examine it under the microscope. In this case, we add a stain which is called the lactophenol cotton blue stain. So we add this cotton blue stain to the wet mount before we cover it with the cover slip, and then we see it under times 40, which is the highest high magnification of the light microscope objective. So you can easily see the spores, as you can see here, as if you see it under the microscope. You can see the type of spores because they are stained by the lactophenol blue stain. And this is one of the easiest methods used in the microbiology lab to for microscopic examination of fungi structure, especially the hyphae, the spores. So this stain is useful for making permanent mount because it sticks the you can leave this slide for a while, for a long time. It keeps its structure in the slide for a very long time. So it's very useful to study the fungi hyphae and the spores. And even if they are yeast colonies, even you can stain them and examine them under the microscope. So this is one method. The other method is what's called a double-sided sticky scotch tape. So in this case, we take a tape, um, الصاج العادي, the sellotape. We take the tape, we call it sticky scotch tape, and then we take a part of the colony from the petri dish, and we put it on the slide together with the stain. It is the same as the lactophenol, but this one is proved to keep the spores intact and the hyphae intact, even their arrangement, unlike the lactophenol cotton blue, because there you use the inoculation loop to take the from the colony, and this might disturb the colony or might disperse the spores. But in this one, you take the tape and then you take part of the colony or very small part of the colony, and then you put it on the slide that has already lactophenol below. So, and then you see it under the times 40 light microscope. You can see again the intact hyphae, the conidia, the conidiophores, the stolons, and so on. So you can study the morphology of the fungi, especially the filamentous fungi. The third type is what we call a slide culture technique, and in this method, you need to grow the. It's usually used to monitor the growth of the fungi. So we need a petri dish, and then we need a cover slide, and then we grow the fungi on a square, like a square or a cube of an agar. And then we grow it on the sides or the sides of the block of or the square of the agar, and then we incubate it with the petri dish in the incubator. Every time we take the petri dish out, and then this is how it looks. This is the cube of the agar, and then we grow we inoculate or culture the fungi in this agar block, and we incubate it. Usually there is a moist. We provide moisture by having filtered paper, moistened sterilized water inside inside the petri dish. So you inoculate it and incubate it, and then every time you take the petri dish and see it under the microscope. This is if you want to study the growth pattern of the fungi, because it takes very long time. And this is usually used by the researchers who used to study or compare the growth or compare the spread or the type of mycelium or the division of the mycelia, the hyphae in different types of fungi. These are the three methods for microscopic identification of fungi. We come to the last part of the session. We are going to speak about the pathogenic fungi of medical importance. We need to know that fungi, they are not commensals, of course, in the body, but usually for example, the candida. We call it it's a type of yeast fungus that we call them opportunistic. So once the body immunity um, lowers or becomes minimum, or if the body is affected by any autoimmune disease, then this type of fungi, they they cause disease, especially in the mouth, in the vagina, in the different bodies where they are found. So unlike bacteria, fungi are large number of them. They can be cause diseases in humans and animals. The type of infection is categorized or divided according to the site of infection or the part of the body that is affected by the fungus. So usually we have two types, which is localized on the skin infection, and this we call them dermatophytes, dermatomycosis, and or systemic infection that infect any other part of the body. but they are further divided. Even those we call them infecting the skin, they are further divided according to which part of the skin they are affecting. So the diseases by fungi are called mycosis. This is the mycosis, and this is the name of the diseases they cause collectively. Singular is mycosis with I. So these are divided into categories according to which part of the body they affect, even in the skin. Is it on the surface? Is it cutaneous or subcutaneous? Or is it systemic? So we have four types of infections. Superficial mycosis are these infections. This is the type of fungi that affect or infect the hair and the dead layers of the skin. Cutaneous mycosis, and these are examples of ringworm, and they affect the skin, hair, and nails. So they go deeper, not only the superficial part. Subcutaneous, and this is the lower level of the skin, and these are affect the skin, the deeper layer of the skin, and the bone. Systemic or deep mycosis, this infect the internal organs, example the lung, the kidneys, the intestine. So there are different infections within inside the body. So these fungi, usually the those that affect the skin, they are transmitted by contact with humans or infected humans or animals. And those that affect subcutaneous and systemic mycosis or organs, these are usually saprophytic fungi. That means they are either inhaled from the environment, and they are all usually growing in the soil. So they enter the body either by inhalation of the spores, or maybe there is a wound or a breakthrough on the skin where they enter into the human body. Uh, some of the fungi, they are not infectious, but they produce toxins, and these toxins we call them mycotoxins. So these mycotoxins are poisonous to the human being if they are ingested. There are certain types of mushrooms زي الفطير، بعض أنواع الفطير اللي بيستخدم في المأكولات. They are poisonous, but usually you have to take care to select the right mushroom. And even those type of fungi that affect the grains, like and it's very common in Sudan, like الفول السوداني, they affect the oily seeds. So they grow because they grow on these seeds and produce like black spots. So this produce toxins, which we call them mycotoxins, and they have other names, aflatoxins. These are very toxic, especially in the peanut of the Sudanese peanut. If the seed itself it is infected. So not all fungi they affect human being, but they might produce mycotoxins which are poisonous to human beings and animals. The table here shows the common diseases that infect human beings according to the site of infection. For example, in cutaneous and superficial mycosis, we have different type of tinea, but they differ in the infected area. For example, we have ringworm, and this affects the scalp, the head, the hair scalp, and um, and this is caused by Microsporium species and Trichophyton species. So there are different type of tinea. This is a common name of the disease, and this is the where the site it affects, and this is the species. Usually, not usually, but most of the time, there could be two species causing the same disease. So you might find different species of the fungi leading to the same disease. We have one of the common diseases, which is aspergillosis, and this affects the ear canal, and it's called by Aspergillus species. In the other type of fungi, they have different sites in the body. They could be systemic, they could be subcutaneous, and this table you have to note it by heart, and you need to know the name of the disease, the where it affects, and the species. This is very important to know and study it by heart. Very important. Thank you, and see you in the coming lecture.